Road, mostly the CBCS Road, but we've managed to kind of break them over here and kind of engage them and doing some stuff with us, which has been an absolute pleasure. So when Brian was coming over, his partner works for the Nature Conservancy and my partner works, and I was talking to Eddie and my partner about it. And his words were, he's one of the smartest ecologists of this generation. You need to talk to him. And you know what, he was right. Brian is an incredibly smart guy, but he's also an incredibly generous and nice guy. And so I'm really happy that he's been able to come and work a bit with my group and hopefully engage with you guys a bit more, because we can certainly learn from the way he's thinking, the things that he's trying to do, and I feel like we can also kind of create some really interesting work by bringing in some of the conservation angle and the different ways that maybe other groups in this school are thinking. So thank you so much, and I'm going to hand over to you.
Thank you, Eve. Oh, Eddie, you're weird. It's great to be here. Thank you for the invitation. Really had a wonderful time interacting with people at CSIRO and here at U2. I look forward to potential collaborations. And this talk is about incorporating ecological theory and understanding into conservation practice and making sure we can get all that we can out of our designs. And the way we're looking at that is taking our understanding of cooperation, mutualistic theory, understanding how that varies in time and space, and then asking ourselves, have we incorporated all the positive species interactions that we can into these conservation? And if not, what happens if you do? Does that change the trajectory? So I want to start off with a little story. I got hooked on this idea maybe 15, 20 years ago. I was in Chile with Mark Pertness, I was just starting my PhD, and we were interested to look at how interactions between individuals change along some kind of physical stress gradient. So we went down the marshes along the Chilean coast, and we planted plants along a oxygen stress gradient with and without neighbors. And we wanted to see the growth. So we set that up and we came back 3 months later. The problem was, during that 3-month period, there was a tsunami that came in and brought sand all over the dunes and then landed on the salt marshes. And so it put about 4 inches, it was a perfect amount of sand on the marsh. And we went back to our plots, we still see the PVC poles. And interestingly, the only plants that survived were ones that we had put in clumps that had neighbors around them. And this was predicted by theory, but the idea is that under high disturbance and high stress conditions, having neighbors is really important and can lead to increased success. In that case, that looked like it was an ecological experiment, but it was similar to restoration. And one of my ideas was, well, what happens if we incorporate positive species interactions into restoration or conservation designs? Does it change the trajectory? I'd like to start with the acknowledgments. This work has been led by students and postdocs. They deserve most of the credit here. And also acknowledge, I'm over here because of the Fulbright. There's an amazing, Australia probably has the strongest Fulbright program, and there are over 200 fellowships now for Australians to come to America. And they're probably have the strongest monetary support of any Fulbrights here. So if anybody wants to come over from the Duke, let me know. The LendFest program and the National Science Foundation for their work here. So the goals of any conservation project are, you have a target of interest and you want to stop the decline or increase the concentration of, for instance, a population, a community, or an ecosystem function. So the idea is identify that target and stabilize it or increase it over time. In marine systems, looking at restoration and conservation, a lot of the paradigm in which we work was taken from forestry science about 40 or 50 years ago. And the idea was that you need to watch out for competition. So keep these animals and plants away from each other as the ecosystem is establishing, and therefore you will increase the productivity and the regrowth of the community over time. Indeed, if you look at or participate in marine conservation projects, there's a discussion like this in the room. A target will be identified, population, community, or function, and then typically you go over thinking about all the threats that could impale or impair that population or target from going back. You look out for physical stressors and try to reduce those in designs. You look out for human impacts, and you do that with protected areas of management of human behavior. And we also think about suppressing negative species interaction. Watch out for competition and invasive species effects. And if you put all that together, the composition really focuses on systematically identifying and avoiding negative interactions that will suppress population growth. But we all know as ecologists that positive species interactions are paramount to these systems for their stability, their resilience, and their growth over time. This is an example of a saltmarsh ecosystem off the east coast of North Carolina, and their expansion on the backside of Barrier Islands is tied exclusively with the formation of these oyster reefs right here on the fringe. So this is a bivalve reef. They start to form, they grow over time, build up land, and they move seaward over time. And due to their building of land and the baffling of waves, they allow an entire ecosystem that's not touching with it to develop over time, and that ecosystem can expand over a couple kilometers. So this positive interaction essentially generates and maintains that ecosystem for occurring. And we know from intro textbooks that there are all kinds of cool positive species interactions out there. There are direct facilitations where A facilitates B. One species can have a whopping effect, like a foundation species, and facilitate an entire community. Of course, there can be feedbacks, and that generates a mutualism. And we also have a series of indirect interactions. You can have evenly numbered negative interactions, 2, 4, 6, that will generate one or sequential positive indirect interactions. And this is a really powerful one I'll get to later on, is sequential positive interactions can have synergistic positive effects that are non-additive that can really generate lots of biodiversity. We also know these interactions aren't just going on with animals that are close to each other. You can also have long-distance facilitation where ecosystems that are kilometers apart are actually affecting each other. We know that from, of course, the Great Barrier Reef and other reef systems that dampen waves and allow seagrasses and mangroves to coexist in that space, and then mangroves and seagrasses can have positive effects on corals by suppressing sedimentation and sucking up nutrients before it gets to the coral reef. So there's long-distance facilitation too. So the three questions I want to go over are: positive species interactions critical for marine ecosystem structure and resilience? Everybody knows, has examples where it's important in your ecosystem or for your population. I'm just going to give you examples from what we have found in coastal systems, how important it is. When and where are they important? This is really important for not just looking at system-specific knowledge, but conceptually, can we predict when positive species interactions should be important in any ecosystem, or different types of ecosystems, or ecosystems experiencing different physical conditions? And experimentally test can inclusion of positive interactions systematically and by design change the trajectory of increased success. So the first story, I had 1 to 3 slides on each one of these stories. We investigated what controlled biodiversity on Patagonia rocky shores. This is one of the most windswept, driest rocky shores there is. Most of the work on rocky shores has been done in temperate rainforest. And Bob Payne in 1966 came up with the idea of keystone predation. And he found by plucking sea stars off the rocky intertidal that there was a huge indirect effect of sea stars as predator in facilitating biodiversity. When you pulled the sea stars off, mussels started to accumulate on the system and clog it up and knock out biodiversity. So the idea was that keystone predation mediated or enhanced diversity. When we went down to Patagonia rocky shores, we did not find any sea stars at all. It was just one mussel, and in fact, we couldn't find any of the organisms that typically occur on the rocky intertidal until we took out our ice scrapers, what good rocky intertidal ecologists do. They do disturbance experiments right away, and you pull out these little patches here and look how fast they close without species interactions, and that's where all the animals were. It was like the dollhouse, a mini-me of the rocky intertidal world. Snails were like this big, everything was like 1 to 2 centimeters, and these are the biggest crabs we could find. This is the sea star, Asterias minimus, a perfect name for that. So we went back and had some mate, trying to figure out what to do with this system, because the keystone predation wasn't there, and all the diversity was in the mussels and it wasn't on the rocks. So we asked the question, what is the relative importance in this highly stressed physical system of predation and direct facilitation and controlling biodiversity, recovering physically stressed systems? So we cleared off plots, and we excluded predators, had cage controls, or excluded predators with mussels and then mussel mimics, and waited 3 years to see how biodiversity accumulated. And this is what we found. This is in the high intertidal and low intertidal, so less stressful. This is bare. And in any of these plots, if you go to one that's really aged, you can find up to 30 species. It's a pretty diverse system. Over a 3-year period in a bear, in the high zone, only 2 species showed up. It was an invasive barnacle and this one species of algae that can survive outside the mussels. And what we found is that there was no effective caging that couldn't be explained by cage control. So predators over 3 years had no effect on this intertidal system. That's the first intertidal system I've ever seen that to be the case. If you shade in a cage, it really doesn't have biodiversity. It's only when you put rocks or mussel mimics or mussels themselves that you get biodiversity coming back. And we do that with the sponge. It turns out these mussels are creating this really nice condominium in there that doesn't dry out. And all these animals, if you tether them, they die within about 45 minutes. They're not true rocky intertidal animals. They can't handle that stress. And so the whole community was facilitated. We call that whole community facilitation by a foundation species under stress. Bob Payne didn't believe it, and this is Peter Grievous that I called us up, and he said, let's go down there and see that. This is a great story about Bob. He was almost 80 at the time. He's unfortunately passed recently. And so we had a whole consortium of students from the University of Patagonia. We went down to the Rocky Shore. Bob had thick Coke bottle glasses, could hardly see. I didn't know how he was going to deal with this rocky intertidal. So we get there after a 4-hour drive, we get to the rocky intertidal about right here, and he gets down on his hands and knees, proceeds for 3 hours to crawl all the way to the intertidal right there. Because you had to hold his arm walking through there. It was amazing. And he said, well, Brian, you're right. Maybe just for So he did believe that there was whole community facilitation, that sea stars were unimportant in the system. In Argentina, we've also found that facilitation is key for population persistence over time. And this is a really diverse 2-species community of plants. So we have this succulent here, and we have this herbaceous grass. And what we found is that because herbivory is so intense, likely due to predator loss in the area, but these herbivorous crabs love this plant, this herbaceous grass, but they do not like this succulent. And so this plant only has a chance if it grows up in this ring and has a neighbor around it and gets big enough so that it can withstand enough herbivory, only occur on its edges. When it's very small, the crabs weed the entire plant. So these started out the same. And you can see this didn't have a neighbor and this did. And you can see it's a different trajectory when you have facilitation occurring. Just a little bit can change the population growth. Everybody has those examples. These are just from our system. So we've been looking at the role of mutualisms in community recovery after massive disturbance. So like coral reefs, forests, salt marshes are also experiencing massive die-off. But unlike most systems, they aren't going into an altered stable state. We see recovery. So we lose, every 10 years, we'll lose 300,000 acres of salt marsh as drought intensifies. So drought increases, we don't have rainfall, that stimulates overgrazing in these areas. We have snail fronts, crab fronts that go through and kill the grasses. Predators die off in the drought. And so what happens is that we get runaway grazing and physical stress leading to massive die-off in the system. So because it recovers, we have asked whether or not the role of positive species interactions in the ecosystem recovery after these massive disturbance events. And so we do our favorite experiment here, and we look at whether or not plants grow better with and without neighbors, and we see a big neighbor effect here. That if you grow plants, these individuals, and you put them out here in this die-off zone, that is very stressful because of low oxygen levels here, is they grow better with neighbors. And the reason is because plants are messy when they shunt oxygen into their roots. They open the stomata, oxygen goes down into the roots, and then it fills up and it leaks out. Microbes take advantage of it, and neighbors can take advantage of each other's leaky roots. And that's the mechanism. So we get big effects of growth. We also found that interspecific mutualisms are critically important and are likely the reason this system is not slipping into an altered stable state as drought frequency increases. So this is a larger picture, a drone picture of a smaller die-off area. And what you'll see, just like in most ecosystems, die-off is not complete. It's not like that little patch in the rocky intertidal. There are all these remnant patches. And it turns out those remnant patches are really, really important because this system closes up through clonal growth, not through seedling establishment. And so their high edge to area ratio means that they contribute disproportionately to the regrowth in that system. So we want to know, why are those patches there? And it turns out that almost every one of these patches, about 98% of them, have mussels underneath. There's a mussel mound, and this rib mussel builds mounds like this. Some of them are as big as this room, and they make sinuously reefs. And about 1% of the marsh surface actually has mussels. But we found, even though they're only 1% of the surface, they determine or associate with 98% of survivorship. So we did experiments looking at above ground plant growth without mussels. They do bump up plant growth by about 100%. They also reduce salinities. It's more like a shower drain, so it's always wet, and the marsh around here will drain through those mussel mounds. And they also increase nitrogen content in there through filter feeding. And in the process, they also attract predators that kill off grazers. So this is it's a stress amelioration zone. It's like strong positive interactions. And if you look at regrowth and do some models and look at the relationship between die-off size and years to recovery, they see with mussels in the spatially explicit model is that no matter the die-off size, we always get recovery in 10 to 15 years. And that's about what we've seen through observations. But you get this interaction in that in small die-off size, it really doesn't matter if you don't have mussels. But it really matters as die-off and disturbance gets bigger, is that it takes a much longer time for the system to close in. And drought is coming about every 20 or 25 years. So mussels, the system closes up before the next drought comes because of this interspecific mutualism. So here's another one from seagrass systems. And we found that seagrass expansion into the tropics and productivity is highly dependent on this clam right here that occurs throughout the world. And the mechanism is it's nutrients, but they also have sulfide oxidizing bacteria on their gills, which is super cool. It's like a tripartite mutualism. And this sulfide, without the clams, the sulfide buildup in the warm tropics kills seagrasses. But clams on the roots, they're fine, and they're super productive. So a question you might ask, why would you replant seagrasses without that clam, especially in the tropics? And that's actually what's done in most cases. Another positive interaction besides interspecific mutualism is one we all know about and we talked about already, is an indirect positive or facilitation generated by sequential negative interactions. And we found in salt marsh systems that with the subsiding of drought, it becomes very important. And in relation to coastal wetlands and in conservation, we all know it's important to understand the paradigm of ecological understanding of that system. And in marshes, in many ways, were the quintessential system thought to be controlled by bottom-up forcing. So as food web theory was developing in Harrison, Smith, and Slovakian from Yale and Hutchinson, the Odoms were coming down in the US and looking at Lindeman's 1942 paper on food webs and looking at energetics and looking at bottom-up control of trophic levels and dynamics and systems. And they came down here and looked, did observational work, and they found that there was really nothing eating the grass. And the idea was that these plants had won out on the evolutionary arms race. And this system represented the quintessential system controlled by bottom-up forces, where there was a severing of connection between top-down food web interactions. So it was considered a tridal food web. But we've done experiments not only here in marshes in Georgia, but around the world, and we found that actually that observation was wrong, and grazers aren't attracted to detritus, they're actually creating the detritus themselves. So when you experimentally test that and remove grazers from the system, this is what it looks like naturally, and if you remove grazers, that's the big effect that you get in southern salt marshes. And we've also found that these snails and crabs that we find in Argentina, that we find in China, that are devastating those marshes, are highly regulated by predators. Not surprisingly, they're popcorn for all these predators that are coming into the system. Through a series of experiments and surveys, we found that predators are critically important for maintaining this system. And as drought subsides, these predators respond to high concentrations of grazers and suppress them. So predators are important. These are three different positive interactions that are really key. And here's another one that we experimentally demonstrated for the first time in ecology in 2007. And this was the idea of a facilitation cascade. And terrestrial systems have been thinking about this for a while, of course, with trees and epiphytes, and we just gave it a new name. And this is a system in rocky intertidal in Rhode Island. And it's on the edge of marshes, range because there's high wave stress. And one plant can withstand that wave stress and will establish itself, and its roots then lay down and hold on to these rocks. And once they do that, the rocks no longer tumble around, and the subtidal animals jump right up underneath those grasses. And the mussels congregate around the roots, barnacles lay on that, and then we get this biodiversity hotspot because this grass is there. We've pulled away the mussels, and all that biodiversity disappears without the mussels. They need something to hold on to. If you pull away the grasses, all the mussels die because they get cooked in the intertidal or they get rolled over by the cobbles. So we found that there is an obligate set of positive interactions that maintains biodiversity. You first have to have this foundation species, the grass mimic, and then you have to have the mussel. And then you get this out of proportion effect on biodiversity, so facilitation cascade. So those are some strong interactions. The next question on the next few slides is asking when and where positive interactions are important. Is through our synthesis, through theory, when we make predictions about where they would be important if you don't have knowledge of that system to begin with, or a little bit of knowledge in that system. So generally ecologists have done qualitative comparisons like this one, where we compare what controls biodiversity on rocky shores, and we compare to a temperate rainforest, rocky shore and a temperate rainforest versus one in the desert. And we found that increasing climate stress, that we shift from facilitation by predators to facilitation by foundation species. So there's different types of positive interactions with biodiversity there. But what about a more general understanding? My advisor and Callaway came up with this very complex model to predict how interactions with your neighbor would change along an imaginary environmental stress gradient. So they drew up this straight line like this. It was actually a happy face hypothesis. This is the least complex. I'll put this one up here. And what they found, what they suggested was that as you increase stress, the drought stress in a system, heat stress in a system, cold stress, that you're more likely, the net interaction with your neighbor is most likely to be positive under high stress than it is with negative. And so people said, well, that's pretty straightforward. That's a good one to test. Let's just go across environmental gradients. And they've done that in a lot of plant systems. And we've looked at synthesis of whether or not that is true or not. And we've seen that it generally holds. This is quite a few number of studies across a variety of terrestrial and marine systems. This is the effect size, whether it's a net positive or net negative interaction. This is with the stress imposed. This is without the stress, and this is how long they imposed it in those studies. And we see when it was a short stress that when you impose the stress, the net interaction becomes less competitive. And then if it's a longer stress, you get the switch here from competitive to facilitative. In general, when you put stress on neighbors, they tend to have more of a net positive interaction than a net negative interaction. So it seems to be something that's general. This paper gets into when and where that might happen. So we do have some quantitative support for the idea that if your system's under stress, positive interactions are likely to occur with neighbors rather than competitive ones. We just did this synthesis for habitat cascades. This is one of my favorite interactions just because it has such out of proportion effects. And we looked here in this plant algal filament interaction, tree mistletoes, and we know the tree epiphytes. These are examples, marsh grass mussels, seagrass, seaweed. In each one of these cases, this secondary foundation species is rare or just can't even exist without this first one. And the second one really creates a lot of complementary heterogeneous space. It ameliorates different types of stresses and it creates an abundance of niches. And we found that this is common throughout all ecosystems. You can find examples. It's something we can potentially take advantage of more in conservation or restoration. And we found that it was more important and had a stronger effect on biodiversity in more stressful transitional systems. And then some trend in freshwater or not. So there was a big effect of these facilitation cascades on biodiversity. And if we compared it to the syntheses that have been done on predator facilitation of biodiversity, and these effect sizes were larger in the literature. We've also looked at ecosystem type and the effect of predator facilitation of plants. And in this case, we found that predators are more likely to facilitate plants and indirectly have a positive effect on their growth in salt marshes. There's a positive effect versus mangroves where we saw no effect. Generally, there are cases. So if you're restoring salt marshes, you may want to pay more attention to the predators in the system. So, positive interactions are important. That's just our system. You guys have all the examples that are going in your system. What if we systematically change our view and say, let's cash in on positive interactions. Here's a flow diagram. So we've done about 5 or 6 of these papers for system-specific ones for corals, coastal wetlands, mangroves, and then we've done general ones as well, papers like that. And then we continue to go out there, and it doesn't seem like designs are changing in those environments, and so we asked, is anyone reading or eating our papers or listening to what we're talking about? And it turns out when you talk to managers, or if you look, only 3 restoration ecology studies, only 3% of all studies in our data set looking at oyster reefs, seagrasses, and salt marshes specifically tested for the impacts of positive species interactions. Even though without them, the ecosystems don't recover or barely recover at all. So very few are testing for that. And we also find that in the designs, they aren't incorporating them very much at all. So it doesn't look like it. People are reading them, maybe not changing what they're doing. So we decided to change perspectives on the applied component of our lab. And we wanted to look at what methods were being done in restoration and then try to change those designs without much money difference and say, can that alter the trajectory that we're seeing? So this is typically how you would plant coastal marshes, oysters, mangroves, and you would separate individuals and keep them away from each other to avoid competition. But our paper suggests that you should clump them and not disperse them. So if you control for density and just look at configuration effects, we did that. We did that in the Netherlands. We did that in salt marshes in the United States on the east and west coast. And this is what we found. Here's the data slide. This is from Florida. This is from the Netherlands. Survivorship and biomass. And again, we did a stress gradient. So we did elevation low and high. And low elevation is more stressful because there's more flooding for these terrestrially evolved plants that are moving back into the ocean. And what we find, remember, they all start out with the same biomass. And the black bar is clumped. So you see there, after a 2-year period in the low zone, we see almost 100% increase in survivorship and a 300% increase in plant biomass. So initial conditions the same, just put them together so they touch. High elevation, we see it's less strong, but still strong. And we see the same pattern in the Netherlands. But notice this is really interesting, is in the high intertidal, we have this interaction, it's very small, it flips. In the high intertidal in the Netherlands, it rains there all the time. It's really not stressful at all. And clumping had an adverse effect. They actually should be dispersed. And so we took this information and we talked to policy managers and they said, really what we need is a schematic. This really doesn't help us. So we came up with this as so it incorporates positive species interactions but also theory. The idea we know they're important, but not in the stressful part of the disturbed environment. And so we suggested the high intertidal, they should have dispersed plantings, and they should be clumped in the mid and low intertidal. Now we're doing this restoration managers. This is actually the key, is a conceptual diagram, bring an artist in there, something that communicates well. And their restoration policy makers that are testing this at a larger scale. We're doing this with clams now and seagrasses. And seagrasses are really taking off. Seeds are becoming effective now in restoration, but we have a lot of failure. And so we asked 2 things, is we put clams underneath shoots that were outplanted and also seedlings. And we found huge effects. Seedlings all die without clams, and with clams they all survive. Not all survive, but the plot gets big and the plot grows over time. And we get a doubling or tripling of growth rate with clams underneath. And these clams are easily available. Many times they're just over here on the sand flat so they can be collected. And you only need to put 1 or 2 underneath those seagrasses. So it has a whopping effect for a small cost in those environments. And we've done mussels and marshes and it does the same. What about trophic facilitation? So one of the things they do on the Great Barrier Reef and on our reef system is they pull off corallivores. And one of the ideas is that with the loss of predators, there's an outbreak of meso-consumers in the environment. And in this case, can this really help with the restoration or maintenance of these systems? So we went in and actually experimentally test whether this intervention actually has the outcome. Because people are saying it doesn't, there's nothing really, local interventions don't help guard against global stressors. So that's mostly observational data, but we want to experimentally test it. So we pulled these, we removed all the snails from these brain corals, we kept some as control, and then we increased their densities as well to naturally occurring high densities. So we had like 0, 5, and 10. Sort of a great undergraduate project design, and very simple, and then we got big effects over a year period. First of all, we found that 5 snails ate about 5% of the coral head in a year, tiny bit, and 10 snails ate about 8%. It was just minuscule. We thought the corals were winning, but it had a huge effect on those ability of those corals to resist bleaching. So we did a bleaching event in October, and we went back and got bleaching data. This is a method that Ove and others here came up with, a colorimetric assessment of bleaching. And what we found is with high amount of snails, we get a significantly different than average or a removal, and this is the amount of bleached tissue. So if you pulled off, if you had high densities of snails, and you went completely white, all of those antennae were gone. But if you had lower numbers of snails, the bleaching was only partial. So they retained those antennae to the next day. And the cool part was the recovery. If you were this, is that's what bleaching looked like, you turned into a basketball of algae. Just 9 out of 10 did. If you were like this, you were much more likely to recover and grow back. And this is what this shows. This is looking, the bleaching severity is high here, and if you have high bleaching severity, tissue mortality is almost 100%. Low bleaching severity, you're likely to return to normal over time. So this is one of the first experimental demonstrations that a local intervention of stress reduction locally can actually increase the resistance of corals to global warming stress and bleaching stress. So that's a positive interaction we certainly should include. And this is one of the last examples with restoration. And this is one of my favorite stories. Brent Hughes is a former postdoc in our lab. He figured this out. It was just amazing. It's in Elkhorn Slough, California, it's the toilet bowl of California. It smells bad, there's rafting, algae everywhere. It's like pea soup. What happened about 14 years ago is sea otters started coming back in. They were beating each other up in the kelp forest. They're pretty mean to each other. And they moved in there and they just started eating everything. They eat 25% of their weight a day. So they'll eat a crab like this and they'll go back down and get an incubator worm. It's like this big, and suck it down in a minute and go get another one. It's just amazing to see these animals eat. And what happened was oysters were, as otters were coming back, seagrasses came back and they're like 8 or 9 feet tall and they're like kelp beds in there now. And at low tide they float on them, so you can see them just bending like this. And he figured out that was because of trophic interactions, and that otters, by eating all these crabs, facilitated sea slugs, and these sea slugs proliferated, even these isopods here, and keep the seagrass blades clean, and therefore they don't get overgrown by algae. It's fueled by nutrients. And check this out. This is amazing. So what happens? We have nitrogen loading, and if you're a marine ecologist, you learn about alternate stable states in seagrasses. And what you know is there's a tipping point that's supposed to happen at about 60 micromoles, and that's when seagrasses disappear because they get overwhelmed with algae in that system. Seagrasses die, it becomes turbid, they never come back. It's too turbid. Can't happen unless you just lower nitrogen in the system too big. And this is where Elkhorn Slough, this is otter recovery, and this is what I want you to look at. Is it's super high up here in these areas. This is where Elkhorn Slough is, and this is where the super productive seagrass beds are. So they're up there, and they're expanding like crazy in the presence of one of the highest concentrations of nitrogen in any estuary. And so this shows us that sea otters are naturally allowing for natural recovery of seagrasses, and now they go plant seagrasses only where there's otters in those areas, and they grow. If you plant them without otters, all the seagrasses die. And so we're getting massive expansion. So here's an example of, last couple slides, what these things can do. So you can imagine you're at a conservation table and you're thinking about, okay, here's our ecosystem, the amount of corals, forests, and here's our physical stress, it's drought or warming. And this is where the threshold in the system is. And typically the way you manage for that, to stay away from that potentially tipping point, is to reduce some stressors, or with a multiple stressor perspective, and say, well, we'll reduce some threats, and therefore they'll be able to withstand, for instance, a global threat. We'll reduce local threats. And we want to stay away from that. That's how we manage that. Well, the alternative we present here is that you can actually expand that threshold if you insert or maintain positive species interactions in the systems. So if you pull off these grazers in the system, you increase the threshold, we know, about 10 to 15% for corals. It's another 40 years or so. So that's a pretty nice positive species interaction. So if you quantify that. If you look at mussels and marshes, their salinity tolerance goes from, their drought tolerance goes from about a negative 2.3 to a negative 4.2 on the drought Palmer index. So they can withstand much higher droughts as long as those mutualisms are there. And so it increases the threshold in the system. And sea otters, that's almost 1,000% increase. It's a huge effect. We had a great question yesterday about what about, does that mean we don't have to reduce the nutrients in the system and the pollution in the system? Well, there's other effects as well. And that would be a fun one to discuss. But the alternative to manage to staying away from the threshold is to expand it with insertion of positive interactions. So this is the last slide before conclusions. Some people will ask, well, what about, this is great that predators are having that effect. We don't have these predators in the system. One of the things we noticed in coastal wetlands over the past 10 years is that we're more and more likely in nature reserves to be stalked by large predators. And this was something that was not supposed to, so we were being stalked by alligators, which are freshwater crocodilians in these salt marshes. And we started to see sea otters, which are supposed to, just like kelps, they were coming into seagrass in salt marshes. In Patagonia, we saw puma in the grasslands, which are supposed to be mountain lions. So we're seeing all these animals misbehaving. And if you start looking at the literature, it's pretty common that after local conservation success, and we do have success stories, is that large animals tend to expand, this work is going in rivers in some places, out of that habitat that we thought they were specialized in, it was their favorite, and they're moving out into other environments. And for many of these, they're doing better growth-wise. Alligators, unbelievably, can deal with fresh water. They just go get a drink of water like we do, and they get back in the ocean and they just have a smorgasbord. There's so much food there. Their growth rates go crazy in these coastal environments. Wolves are coming down. So there's a lot of ecosystems out there that have an opportunity for positive feedbacks from predator expansion, not just the sea otters. So what we're doing now, and this would be fun to collaborate with people here, is asking what happens when you layer positive species interactions in conservation design? What happens if you bring them into fisheries management and do self-organization with communities and self-organized seagrass beds? And then if you layer those, do you get synergistic, I doubt it, but maybe get some additive effects? Or some other things that would really bump that. How far can we go in increasing the resilience of these systems if you layer it? Certainly, I mean, with sea otters, clams are important for the seagrasses, but sea otters, they obligate to one, but you add clams or clump them, they actually grow better. So positive interactions can increase restoration success and increase system diversity. They're important in all the systems we study. I'm sure they're important there. And for marine conservation, we really need to systematically list all the positive species interactions. So for coral reefs, we can't just say we have to have parrotfish. Kenny gave a great talk and he said these cleaner fish never get talked about in conservation, but have potentially really strong positive effects by affecting the density of herbivores. So we need to systematically look at those. And then the likely result is that we're going to lower conservation costs while enhancing system resilience. So hopefully a little optimism that we untapped efficiency in conservation. So thank you very much.